And if you're told that they are actually sister taxa, they belong to sister taxa, you can then intuitively understand that they're related by a recent common ancestor. And so they shared a genomic history in the recent past. But very little is actually known about the genomic changes that have occurred that actually create the split, the actual process of speciation that creates the split, leading to the two different lineages. So whenever anyone gives a speciation talk, I think they have to have this quote. So this is a definition of a species under the biological keep species concept. And it defines species as groups of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. And so the key idea here, then, of a species is the idea of reproductive isolation. So really when we're asking how do new species evolve, what we're asking then is how is gene flow being prevented? And here on the bottom I have steps that are required to happen in order for populations to have gene flow and maintain a population integrity within a species. So if you have barriers that arise that block gene flow at any one of these steps, you can get a speciation event. So for example here, there's two flower types here, and they have two different pollinators, a hummingbird and a bee. And so these flowers won't actually mate because of the differences in pollinator specificity. And so here, this is the point at which gene flow is prevented at the mating step. And then everyone's favorite example of post-zygotic reproductive isolation, the horse and the donkey, they actually get along quite far in the process. Mating occurs, a zygote is formed, a hybrid is even produced, but then the hybrid is sterile, the mule is sterile. So here, this is where gene flow is prevented. So really what we're studying then is the genes and the regions of the genome that are important for barriers such as these to arise. So this is what we're talking about when we're talking about speciation genetics. And so, so I was going to say, and you can have this happen so those were just two examples, but you can have this happen anywhere along the process here. And it's puzzling though, because speciation genetics is actually a really old topic. People have been interested in this for a really long time, but there's many unanswered questions. And these are just some of them. There's many, many of them, but these are some of the hot topics, I guess, right now in speciation genetics that people want to know. So it would be interesting to know the relative importance of pre-zygotic versus post-zygotic isolating mechanisms in early speciation, which ones predominate, what evolutionary forces are important in the early stages of speciation. Is this maybe driven by selection, or is it completely by chance or by drift? What's the genomic distribution, actually, of the reproductive isolating mechanisms? Do they cluster genetically, cluster together in the genome? Do all the genetic components that produce an isolating barrier cluster? Or are they evenly spread across the genome? What's the actual role of gene flow in speciation? And this is particularly important in early stages of speciation, where you might have an incomplete reproductive barrier, or reproductive isolating barrier. And so, since gene flow would oppose the speciation process, how could you actually get speciation in the face of gene flow? Does speciation occur from divergence at single or multiple loci? And finally, do reproductive isolation genes, or so-called speciation genes, tend to be the same targets across multiple taxa? And so it's not that we don't know anything about speciation genetics, and actually over the past few decades we've found some really, really cool examples of speciation genes. And this is a table from Prescribe's Nature Review paper, where he's listed actually all of the speciation genes that have been found so far in all those different systems. And so the first thing to notice here, though, is that this all fits on a table. And it's quite small. And I'd like to point out why I think this is. And so the first thing I'd like to point you to is the species column. And here you can see that it's focused only on model species. So there's Saccharomyces, Arabidopsis, Drosophila, particularly Drosophila melanogaster, and there's a mus example there. And then secondly, if you look in the phenotype category, all of the phenotypes of these speciation genes are post-zygotic isolation barrier, or affect post-zygotic isolation. So only sees sterility, lethality, and inviolability as phenotypes here. And this is most likely not the complete picture here, I would argue. And I think the reason for this is mainly because of the focus on model species, which makes sense because it takes a lot of resources, a lot of time to develop the genetics for one of these species to use in genetic studies. So we use what we had, and so those were these species. But what's not pointed out in this table is that these species comparisons have quite long divergence times when you're comparing them. So the two yeast species that they use, Saccharomyces baianus and Cerevisiae, have a divergence time of 14 million years. Drosophila melanogaster, the workhorse of the Drosophila genus, and its sister species Drosophila simulans, has a divergence time of 2 to 3 million years. And the mus species, semi-species actually, that they have used in the European house mouse comparisons have a divergence time of 350,000 years diverged. And then finally, the Drosophila pseudoobscura races that they have been studying have a divergence time of 150 to 500,000 years old. And so we're getting better here. So we have some more recent comparisons in the thousands of years. But again, this is still, we're talking about hundreds of thousands of years. So this is a really incomplete picture. And I sort of alluded to this in the previous slide, but so this is the reason why we don't know more. We need many, many more comparisons in order to get a complete picture of what's going on. Likely, there are different things going on in different systems, so studying a lot of different systems would be a good idea. Also, model species might not be ideal for studying speciation. They might have long divergence times. And so, so this figure here is famous. It's from Chung-Yi Wu's paper on gene speciation. And he points to the need to look at populations and species in the early stages of speciation, or early stages of divergence. And he points out, so he's showing here a diagram of speciation through time and what it might look like throughout the genome. And so in an early diverging comparison, you might see a lot of gene flow between the two populations with very small peaks or islands of divergence between them. And under those islands, you might find the mechanisms or the genes that might be important for the actual speciation event. But as you go along and divergence increases, as the speciation time, you get further and further away from the speciation time point, the divergence will increase spread throughout the entire genome. When you finally get to the end here, and you're studying two very diverged species, you may find a reproductive incompatibility gene here, but you have no idea whether the reproductive incompatibility gene you find here is actually one that was important way up early on in the time point. So it's important to study these earlier cases because the differences that you find are more likely to be the ones that are involved in the speciation event because there's been less time to accumulate further secondary changes in the genome. And so in order to do this, though, obviously we don't have all of the genomes to study this, so we must develop genomic tools in non-model species that are more relevant to the topic of speciation. So luckily we're in the age of next-gen sequencing, so we can do this. And I realize this is the vertebrate group, but I'd like to say that I think the Drosophila genus is actually a really great place to start, or at least to do speciation work, actually. Because, so when people think of the Drosophila genus, they just think of melanogaster, actually. But the Drosophila genus is made up of over 1,500 species. And so you have access to many, many, many time points that you can pick. You can pick your time points, basically. And also, we can't ignore that we can actually utilize the genetic tools and techniques that we've developed in Drosophila melanogaster, and then already have a head start at developing non-model systems for use in speciation. So I'd like to point you to this obscura group here, and particularly to this group right here. This is the Drosophila Athabasca group. And this is the group that I've been studying. So this is a little bit about the Drosophila Athabasca system. So like I said, it's a species complex. It's made up of 3 semi-species. There's the western, northern, the eastern A, and the eastern B. And they're morphologically identical. but they have distinct geographic ranges. So you can see on the map here, the western northern has its range across almost the entire northern half of North America, eastern A over on the Great Lakes, and eastern B over the New Jersey-ish area. And so what's also cool about the ranges is that there are small areas of overlap between them. So there's an area of overlap here between eastern A and western northern, and then an overlap here between eastern A and eastern B. And what's cool about that is you can actually, there's potential for gene flow, so we can actually study what's going on in a speciation event according to gene flow. More importantly, though, this species has a very, very recent divergence time. So the estimates of divergence time within the group range from 5,000 to 23,000 years. And this is much, much less than any of the species that were on the table that I showed you earlier. And so the divergence time between the western and the two eastern groups is more ancient at around 20,000 years, and then very recently, around 5,000 is the estimated split time for eastern A and eastern B. And they still produce fertile hybrids, but they exhibit prezygotic isolation in the form of courtship song differences between semi-species. So courtship song in Drosophila is known to contain species-specific components. This is an example of a single pulse train, a waveform of a single pulse train in a melanogaster song. And so the courtship song is made up of different components, and I've listed some of them here. But the most important one that's been known to be important in Drosophila species is this interpulse interval. And the interpulse interval is known to be species-specific. So it's very characteristic of a particular species, and here's just some examples. So the Athabasca group actually, the way that they discovered that they were semi-species was purely by courtship song, actually. They had no idea, and then they discovered there was this prezygotic isolation, and that there were different morphs with different courtship songs. So we had to measure this then with our flies, and so what we did to measure the interpulse interval is you can measure the pulse train length and then divide it by the number of pulses, and we can do this for 3 replicates. We did this for 3 replicates per strain, and you can see that eastern A has evolved a much higher interpulse interval compared to eastern B and western northern. And so you might think that, well, how do they distinguish eastern B from western northern, because it looks like they have the same interpulse interval. And if you remember in the map, these actually don't coexist in nature. So if you find a fly that has a 10 millisecond interpulse interval in New Jersey, you know that this has to be eastern B then. So it's a combination then of interpulse interval and geographic range that will define the species, the semi-species size. And so this is the box that we stick it in. So you just stick a mating pair in a little cage, and you stick it on top of the microphone in this box, and you record them. So I have a little video first of what this looks like. So that's what it looks like when they're singing. This is actually recorded in that chamber that I showed you.
It looks like they use the wings.
Yeah, so it's a wing vibration. And so then I want to play for you the songs from the different semi-species. So this is the western northern. Why is it doing this? Okay. So that's the western northern. Here's the eastern A. And then here's the eastern B. So these definitely, they sound very distinct. And so you can see also when you zoom in, the E2A has this very different pulse structure to its song. So what can we then learn about speciation from Drosophila athabasca? And these are some of the first questions that we decided to look at. So we wanted to look at how the genomic landscape of divergence looked like in this system, which is a young system to look at, which would be a unique new thing to look at in speciation. What forces are then driving the divergence between the semi-species, and then also can we detect particular genes or targets of the genome that are responsible for the divergence of the semi-species. So our first major limitation that we run into, though, of course, is that we have no genome. So the first step was to actually create a genome. So we spent a lot of time actually making sure that we had all of these different library insert sizes so that we could get the best possible genome that we could. And so we made Illumina libraries, and then we assembled a genome de novo, and we also included sequencing of some RNA-seq libraries so that we could get a good annotation, because what is it to have a genome and want to say something about speciation but not be able to say anything about the genes? So we made some RNA-seq libraries, and this is also a benefit of working in Drosophila. So we used experimental evidence to find the genes using the RNA-seq data. We also did normal de novo gene finding methods, and then we were able to use comparative methods with other Drosophila for already sequenced like melanogaster genes. So comparative methods to find genes that way. So we were able to get a really nice assembly. Our genome size ended up being around 150 megabases, which is within the size that you would expect for Drosophila. The scaffold N50, which is a measurement of assembly quality essentially, was 83 kb, and so this is the size at which 50% of the genome is contained in scaffolds. This size or larger. And the average coverage over the genome was 30x. We have 13,000 annotated genes, and when we look at the core set of conserved eukaryotic genes in the genome, we find that you can find 94% of them are complete in the Athabasca genome, and 98% of them are contained partially or complete in the genome. So we were really happy with this, and we wanted to start out first with a really good genome so that we can actually say some stuff about the species. So it was good we have a reference genome, but in order to say anything, what we really want to know is what's going on within and between the semi-species in order to say anything about the forces going on, what's driving apart the semi-species. So what this means is we need a population genetic data set. And so this means we need more flies. So the summer after I arrived here and I started this project, then I rented a van, and I had a net and some rotten bananas, and I drove across the country and just collected flies. Luckily, they live in a nice habitat. They're forest animals, so I got to go to some really cool places. But this was a five-week journey. And so over the whole course of the collection, I actually collected 800 flies and established isofemale lines from them. And then we genotyped them using, instead of keying them out, we just did Sanger sequencing on a single mitochondrial gene. We typed them into species groups, and we found that I collected 400 Athabasca lines. And then I had to classify them into semi-species, so I classified them by courtship song interval. And then finally I sequenced 28 of these lines at 10x coverage, and this was the breakdown of how many per semi-species. It's not equal because I actually did these two last steps simultaneously, and I just hoped that I got an even number, and I kind of did. So then how do we go from flies to actual genetic data? So instead of assembling each line separately, we can take use of the reference genome and just align the reads from each sample to the reference genome, and then look along the reference genome to see where the sample differed from the reference, and then get a set of SNPs for each sample, and then analyze the data that way. And I'd like to point out we ran through a control set through the same pipeline that we used to do all the samples, and we came out with a false positive rate of less than 1%. So again, we were really happy with this, and then we can move on to start asking some questions about the species. So first, another thing that we had to do was to actually, I said before that the semi-species were actually completely behaviorally defined prior to this. And so we wanted to make sure that these actually, if we're going to study the genomics of these organisms, if they don't actually form genetic groupings according to the semi-species that you're classifying them in, you're really not answering the question that you want to be answering. So we first had to make sure that the genome-wide data followed some sort of genetic grouping that you would expect. So first we created a neighbor joining tree, and you can see the samples fall out very nicely. All of these samples within a semi-species form monophyletic groups. You can also see the different time points. The two eastern groups are sister to each other, and the western is further out. We also then did a principal component analysis. It's hard to see the green, but the eastern bee population clusters here. And so you see very nice, distinct genetic groupings.
Principal components of what?
Of the SNP data. Oh. Yes. So you just give them all the SNPs and see how they fall out. So then, and then also we wanted to confirm the demographic parameters. Are we actually looking at a young species? And so here I used the program DADDY, and I came out with divergence times for the western versus eastern split time of 16,000 years, and the eastern A, eastern B divergence time at 6,000 years. And so this is within, they said before it was estimated using mitochondrial DNA that the split was 23,000 years and 5,000. This is within the confidence intervals of that. But I'm going to use these numbers now, from now on, the rest of my talk. So we can also then just look overall at the patterns of the data and see if they conform to the previously proposed model for how this species has diverged. So it was previously proposed in the 90s that this species started out ancestrally like an eastern A population, and then as glaciation retreated, the population spread out, forming the western-northern group, and then later on, more recently, a small population within eastern B broke off due to some sort of founder event, and formed the eastern B group. And so you can see that the patterns of nucleotide diversity and population differentiation are consistent with this model. So eastern B has a very low nucleotide diversity, which you would expect with a founder event. And then population differentiation between eastern A and eastern B is much lower, consistent with a more recent split time. So again, we then wanted to know how we have all these SNPs. What does the genomic landscape of these mutations look like across the genome? Are all the divergences clustered in islands in the genome, or are they dispersed throughout? And then also, since we have access to these multiple time points within the species group, we can look to see whether it follows something like the Chung-Yi Wu diagram of how species divergence occurs throughout time, where you can see a different landscape depending on different time points. So I should also say that in order to characterize SNPs into ancestral versus derived, we had to have outgroup information. So we also sequenced the sister species, Drosophila algonquin, and then we got the finished genome from a collie. And so with that, we were able to find 1.7 million SNPs that are variable within Athabasca that also have polarization information, or ancestral trait information. So what's really cool about this then is you can also break down the SNPs into categories of private and shared mutations within Athabasca. And what's cool about that is then you can know then along which branches the mutations must have been fixed. So for example, the fixed SNPs that are private to western northern, you know they had to be fixed along this branch. Same with this, so the ones that are private and fixed in eastern A, you know they have to be fixed along this branch, and so on. So then these SNPs in this category here are really interesting because we can look to see basically what does the divergence that's unique to these particular semi-species, how is it distributed along the genome? And so when we look at the older divergence time point, the one that involves the western species, we can see this is what divergence looks like. So on the x-axis here we have genomic position, and it's just divided up into chromosomal arm. And then the y-axis is the number of fixed SNPs. And so you can see there's not really a pattern besides, some of the chromosomes have more divergence than others, which is interesting in itself. But really you don't see any very strong evidence for an island of divergence. So, but then when you look at the two eastern species, which have a much more recent split time, you actually see these striking peaks, which correspond then to clusters of fixed SNPs in the genome. And so this is what somebody would call an island of divergence. And so that we were excited by this. We could see, there definitely is a different pattern in the younger species versus the older species, and they do seem to be clustered in certain regions, particularly in the younger species, or semi-species, sorry. So it would be, then we wanted to know what forces might be responsible for driving the divergence in these semi-species. We see these peaks, but what is actually what's forcing them to be driven to fixation in these areas? And so we wanted to know, is the divergence then driven by selection? So a little bit of theory behind what we would expect to see in the genome under a selective sweep is if you have an ancestral population and this red mutation arises that's beneficial for population 1, that will quickly fix along with all neutral variation in that region. And then the same for the population 2 with the blue mutation, which is beneficial for that population. That mutation will fix along with its neutral variation in that region. And so then when you actually scan across the genome, what you'll see is this plot down here where in a region where a selective sweep has occurred, you would see a peak in between semi-species divergence. So you're comparing these two genomes and they're really diverge. But then you see a dip in the within semi-species polymorphism. And so if you're lucky, then you might look in your genome annotation and see, what's actually underneath this peak in the genome, and maybe there's a gene there, and that could be responsible for something that's driving the speciation event in your species. So I made this plot then, the plot in the lower right across the entire genome for all pairwise species, semi-species comparisons. And so this is the older time point, the 16,000 year time point. And so the black peak, the black line is the pairwise divergence between semi-species, and then the red, blue, and green is the polymorphism within semi-species. So here you can see, again, it's just very messy. You don't really see anything, but that's what it is. So then we wanted to look at the eastern A, eastern B, the younger divergence time, and you see there's very, very little divergence, but you do see these peaks here. These are black. So you see peaks of pairwise divergence between the semi-species in eastern A and eastern B with very low levels overall. And so while this gives you a really good picture of just generally what's going on in terms of selection, it doesn't really give you a good idea of exact outliers. So another way to look at this is, again, to use this same method, but instead to break up the genome into equal-sized windows. And then basically, so this is my equal-sized window, and I do this across the entire genome. You can get an average value for each of these things in each window, and then plot them in a scatter plot. And then you would see some sort of correlation, but an outlier, a window that maybe has undergone a selective sweep would be an outlier in this plot, with very low within semi-species polymorphism, but high between. And so we made this plot for the different comparisons. And so this is just one example of the comparisons using the western northern semi-species. So this plot actually looks the same for all comparisons using western northern, whether you use pi within from either. So if you make this plot for pi between western northern eastern A using pi within from western northern, it looks identical to the pi within using eastern A. So, but the general picture here you get is that there's just a blob. You don't really see an outlier. Maybe this gray was not the best choice of color. So really, it's just a cloud of mist. But what's really cool is, you cannot see this. What's really cool is then when you plot the Eastern A and Eastern B data, you actually, so there's stuff here, and there's stuff here. And there's not anything anywhere else except for there's one point there and one point there. And so this is definitely an outlier, and you would expect that this window then has undergone a selective sweep in between these populations. And this is in the 6,000-year-old divergence time, which is super interesting and super young. So then we want to know, there are these regions of divergence that cluster in the genome. Maybe they're potentially driven to fixation due to selection. What can we find out about particular genes or targets that are responsible for the divergence of the semi-species? And so what I didn't tell you when I was going through all of this is that this peak here that has a the cluster of fixed SNPs, this peak here that has the increased divergence between semi-species, and this window here, these are all the same window. And so there must be something going on here. And actually, if you zoom in here on this window, you see the very characteristic, just like the cartoon version of this plot that I showed, where you have the really high DXY between semi-species, but virtually zero polymorphism within. So this is really cool, and this all led up to like the very most exciting blast result of my entire life. So you look to see what's under here, and there's actually a gene. And it's this gene called Nona. And so we looked at, so there's this thing called Flybase for people who don't know about Drosophila, and it's a database of all of the genes in Drosophila, and has a lot of genomic resources. So you can look up what this gene actually does in melanogaster. And it's a courtship song gene. So there was this paper that was in 2001 where they looked at melanogaster and Virilis, two other species, and they actually took the gene from Virilis, put it into a melanogaster fly, and the song sounded like it had the interval of the Virilis fly. So it's known to be contain species-specific information, which is really cool. So we already know then, based on the scans, that this region is undergone a selective sweep in the eastern species. We know that there's an island of divergence here, but yet, so genome scans are good for that, for finding a candidate gene, but we need some other sort of methods to actually find out what's going on in a branch-specific way. So we'd like to know, is something happening particularly maybe in the eastern A group or on the eastern B group? And so we need another way of looking at selection. And so this method was developed actually in Rasmus Nielsen's lab. So this is this is a figure from the human Tibetan genome paper. And so they use this method to find a gene that's related to high altitude adaptation in Tibetans. So what they basically did was they calculated branch lengths using FST values, and then they looked for outliers. So they looked for branches then that were much, much larger, had accelerated evolution then compared to the rest of the genome. And so you need 3 species to do this, which luckily we have 3 species. So what you do here then is you can calculate in order to calculate a population branch statistic for the eastern group, you would calculate the branch length for eastern A to western northern, calculate the branch length from eastern A to eastern B, subtract out the western northern eastern B branch, and divide by 2, and then you're left with the eastern A branch length. And so you can do this for all of the different semi-species. And then you plot then the branch lengths for each of the semi-species. So each plot is for the different semi-species. And you can also calculate then, you can average each of these points and get a genome-wide average of a tree. So this is the genome-wide average of the branch lengths for the tree for all genes in the genome. And if you can guess, so there's this purple dot here, and the purple dot then corresponds to the Nona gene, the one that I just showed you. And you can see what's really interesting is within Western Northern, it's very within the genome-wide average. Also in Eastern B, it's within the genome-wide average, but then it's an outlier in Eastern A. And so what does the tree look like then if you just plot the branch lengths for this particular gene? So this is what it looks like, and there's rapid accelerated evolution along the Eastern A branch. And if you remember, Eastern A is the one that has the diverged intervals interval. So this was really exciting. So finally, what have we learned so far about this? This is ongoing, but so far what we've learned is that the genomic landscape of divergence within Athabasca varies by divergence time. So Eastern A and Eastern B is in the earlier stages where you can actually find peaks in the genome that correspond to divergence, whereas the Western versus Eastern comparisons maybe are more further down in the process, where it's harder to actually see what's going on. So again, it's easier then to tease apart the genes that may be responsible for population divergence or speciation in the rest of the genome earlier on in the speciation process. Genome-wide scans are great particularly at looking at these young diverging populations, because you can actually see these peaks that correspond to the very small sections of the genome that are undergoing divergence. And it's a good way to then determine candidate genes. And then another exciting thing that this might point to is that speciation might be targeting some of the same genes over and over in different taxa, because as I showed that it was already known in different species that Nona was involved in portrait song differences. So that's. Any questions?
That was great, but you didn't say anything about the edges. What did you learn about contact zones between these forms?
Yeah, so we're still looking at it. I think that it actually might involve, we only sequenced 28 of the 400 lines that we had, and this was first actually just, we thought, well, let's just see what we can get. Maybe this will have to be a preliminary analysis, maybe this is all we need. But I think actually in order to look more at the contact zones and look at introgression across the contact zones, we would probably need a deeper sample of the, yeah.
Do you know where are most oldest needs that you found, like in floating regions, cis-regulatory regions? What's the proportion?
I don't remember off the top of my head, but actually in the Nona gene, I know that they're actually almost all in the 3' end, right, within 2 kb of the 3' end of the gene. So it could be mostly regulatory, but yeah.
That's a great talk, everything. Next question, whether anyone is planning mating trials with you?
Yeah, so when they initially worked on these species in the 80s and 90s, they did do some mate choice experiments, and they always found that they would prefer to mate with their own semi-species.
So I think this is a great talk too. My question is about your field trip. So when you captured the Drosophila, did you get only Athabasca? Didn't you get a bunch of?
Yeah, so I got 800 of them, but I actually narrowed it down kind of because I know that Athabasca is a black pigmented fly.
I see. So you could pick them out.
Yeah. So I would kind of I wanted to also, just out of curiosity, I was interested in knowing, what is out there. So I kind of would take a few of other things. But I didn't focus on it because it was hard to, going across these agricultural checkpoints with this many flies in your trunk. It's a bit sketchy. I get really nervous too. They're like sweating. Like, do you have animals? Like, no.
Turn off that video.
You might be able to leverage that for some help with genomics. This video.
Well, it is amazing what you can accomplish these days. I mean, I'm just stunned at the sheer volume of data that you gathered.
I'm just curious because, you know, if you look at some of Patrick Nosel's stuff and some of the bird work, it seems like they're promoting the island idea, islands of divergence, yet you didn't find that. Do you have any ideas that are Drosophila related thing, or we just don't know enough about these early events?
I think you could consider maybe that peak of divergence as an island. Yeah, but perhaps it's so early on that you just see this one sharp peak.
I did have a question actually. So do you think it's sort of the relationship between, you saw different patterns with the really young divergence event versus the slightly older divergence event, but those divergence events are really young compared to what we might expect to see with vertebrates. And I'm wondering if you'd expect that pattern, that sequence in other taxa, maybe in other animals, and if you would expect it to be somehow correlated with generation times. Like what are the generation times with these Drosophila?
I think there's about 10 generations per year. Yeah, I have no idea. I mean, I think it probably does, it probably varies greatly with species. But I think that's why it's important that we do speciation studies in a lot of different animals. I mean, there's probably also not one clear answer to like how does speciation happen, but that's what, yeah.
Karen, do you think that so the history of the group is that you had the separation of the Eastern A and Western Northern, or Eastern and Western Northern, but then you had the separation of those two within them. But do you think that the song difference was not involved in the initial speciation event? Like, can you say anything about how old this sweep is, or?
I have no idea.
Okay.
Yeah. I know that, so the ancestral song is more similar to the Western Northern and the Eastern B song. So it probably had to flip back in the Eastern B.
Or maybe that's
it's just recent.
Yeah, it could be, yeah.
I have another question. So when you look exactly at that Nona gene, I mean, are there a lot of substitutions there, or is it one or a few?
No, there's a lot. In order to get that really big peak, there's like 20 mutations, fixed mutations in that region. So.
Is there anything known about the ultrastructure of the Nona gene? I mean, functionally where they are in the protein? Are they on the membrane, or?
I don't think anybody knows. Not that I'm aware of. We're currently trying to do some functional studies now too, so it'll be cool.
Does it make sense with the timing of divergence to have 20 new mutations, or is that? I mean, what's your idea about where that diversity came from?
I mean, I think that the overall number of substitutions in the genome, when you look at just the number of SNPs, it's actually very low. It's just that they're clustered really in that one region. So it seems like a lot in that one region, but overall the diversity is actually really, really low. So I think it makes sense, but it's just like, it happens to have all been in that one region, which is really interesting. And be interesting to know if all species, when speciation events happen, like they just all cluster in one region.
I guess what I'm wondering though is like, with your basic assumptions about per site mutation rates per generation and the time of the divergence event, does it, like, can you reconcile those all being mutations?
It's 60,000 generation. So it's a lot. It's actually quite a long. It's quite a long time to.
Well, there's also a debate, or not debate, some new ideas coming about fluctuating population size, and at the peak during summer or whatever, that they have this enormous mutation ability, whereas they crash in the winter time and things like that. So it's not the standard neutral model of population genetics, with a equilibrium population size. So things that people haven't really talked about much can be really wild things happening for natural selection to grab purchase on.
So any further questions for Karen? Let's thank her again for really excellent.